kicking sodium out of your cells and bringing potassium into your cells. Why is that important? The reason that's important is it's changing the osmotic balance of your cells. Osmosis occurs when you have differences in concentration of things across the membrane. Right? Why is the cell doing this? Well, if you think about it, your cell is full of proteins. It's full of all kinds of things that are not present outside of the cell. The osmotic pressure of the cell, if we don't do something to balance it, is very high. The cell is trying to balance the osmotic pressure by altering the concentrations of sodium and potassium ions. It's balancing osmotic pressure. If the cell doesn't balance osmotic pressure, the cell absorbs water and explodes. One of the reasons you are constantly needing ATP energy is to keep your cells from exploding. Okay? If you don't balance the osmotic pressure using this pump, your cells will blimp up and they will die. Okay? So it's important that your cells have a continual supply of ATP to help balance that pressure. It's kind of cool. Your cells, most cells around in a multicellular organism have to do exactly just that. Okay. So here's the covalent modification. As a result of the covalent modification, it changes from kicking sodium on the inside to the outside. When you get rid of the phosphate, it kicks potassium from the outside to the inside. So the covalent modification is changing the activity of the protein. One state to the other state. One state to the other state. Like a ping pong match. Back and forth. And back and forth. Yes, sir? Okay, so it is, I'm not sure if I understand what you mean by constantly, but it is constantly going from this state back to this state. This state to, so it's one of these for one of these. One of these.
What is the bottom right state?
What is the what?
What is in the bottom right state, the phosphate state?
Here,
To the left, at the top right. It is pushing out DNA in more than one time before it reverts back.
No, one time each. So one time to kick 3 out. So it does all 3 at once. And then over here it does these 2 at once. Okay? We'll talk about this later when we talk about membranes. But this is an important thing. This is a critical thing for our cells. We don't balance osmotic pressure, our cells are going to burst. It's a scary thought. Okay. Let's see. One more thing, and I think we'll call it a day. Here's the glycogen thing I talked about. And we'll talk about this later, so don't worry about the details on this right here. All right? I simply show you this figure to illustrate what's happening with differences in phosphorylation. All right? We're not going to worry about the details about all the different things on here. When we think about glycogen, glycogen is a polymer of glucose. Our liver is full of glycogen because it means that it's a storage form of glucose. When we need glucose in the situation like I described to you, the liver gets a signal and it says, oh, the muscles need glucose. I'll break down some of this glycogen, and it releases glucose out into the bloodstream. Okay? So that is important to control. This process of signaling means that the liver has to be able to respond very quickly to the needs of the muscle cells. If it doesn't, then this person with these muscle cells might get caught by some organism. Or it might collapse, or it might have some big problem. So this need is very quick. It needs to make these things happen. So what does it do? Well, what we see is that this enzyme up here, okay, which is called glycogen phosphorylase, this enzyme breaks down glycogen. And it exists in 2 different covalent forms. One form that has no phosphate, and one form that has 2 phosphates. And no, there's not an intermediate. Okay? One versus the other. The form that has the phosphate on it, okay, is the more active form because it readily flips down here into the R state. This happens very easily. Whereas the form that has no phosphates, this doesn't happen very easily. It mostly stays here in the T state. So you have T state, R state, T state, R state. When we put a phosphate on it, it favors going into the R state, essentially. When we take the phosphates off, it favors staying in the T state on top. Now, this means that putting a phosphate on is a covalent modification, and so is taking a phosphate off. Putting a phosphate on is a modification. Taking a phosphate off is a modification. The enzyme, and we're not going to worry about names for the most part, but I want to give you a couple simple names that I think you should know. An enzyme that puts a phosphate onto something is called a kinase. An enzyme that takes a phosphate off of something is called a phosphatase. Those two names we'll talk about later. So both of those enzymes are involved in covalent modification. Kinases put phosphates on. Phosphatases take phosphates off. Okay, I think that's a pretty good mouthful of stuff today. You guys are looking tired. I'm getting tired. Let's call it a day. If you have questions, oh, by the way, I told a couple students earlier today, you are welcome to call me if you have questions. I'm going to give you my number. You're welcome to call me my cell phone. You can call me any time of the day or night. I turn my phone off when I go to bed, so I can assure you you won't bother me if you call too late. Okay? If I'm answering my phone, you can assume everything's okay. My phone number, 541-908-4465. Feel free to call me. Okay? Study hard. Kinase puts phosphates onto things, and a phosphatase takes them off.
Oh,
okay. Hi.
I just wanted to make an appointment with you to come in, possibly,
Sure.
and I have some questions. And also, I'm applying to medical schools, and I know
good.
you're an advisor.
Yeah, I'd be happy to talk
My
to you.
name's Amanda, and I talked to you a few weeks ago, just on the phone.
Sure.
And I just wanted to ask you a few questions about that.
Sure, I'd be happy to. Yeah.
Script.
Sure.
I know. I looked at your.
What's your major?
Biology. I actually graduated from another.
Okay,
school.
you're a postbac?
Yeah, and
Good
I'm just taking a few more classes that, to finish it up.
Okay.
So
here's my schedule if you want to look.
Okay.
Let's see. So you want to come in tomorrow?
I have class most of the day tomorrow, so maybe Thursday. I know I looked at your schedule and you don't have anything like around 2-ish.
That would probably work. Yeah, I think so. Let's see. Oh, actually, Thursday. Now, the problem with Thursday. Let's see, get the day schedule. The problem with Thursday is that this meeting is going to take 1.5 hours to 2 hours.
Oh, okay.
So I'm not sure. I've got somebody coming by at about 2:30. Let me think about it. What was the deal with that? I think it's a student in the class. So I'm planning to be back by 2:30. So if you wanted to come at 2:30, we could at least talk about the class things. I could answer your pre-med questions if nobody else is there then, or we can talk about that separately.
What do you have tomorrow? Maybe that might be
tomorrow? Sure. So let's see. There we go. Tomorrow. So I teach at 9 in the morning. I've got a meeting at 10. And I've got a meeting there that's probably going to last an hour or so. I could meet with you. See, I've got a student coming in actually from this class at 12:30. I could meet with you then if you'd like to. Or if you'd prefer, I can meet with you at 1:30.
1:30 tomorrow, I think would actually work.
Okay. Do me a favor, send me an email, and that way I'll get it on my calendar.
Okay, great. Thank you.
Okay,
I'll
sure.
see you
Hi.
You're so lucky.
Well, I wish I were. So, what's up?
For these two, do we need to know these calculations? Because I was thinking that I guess I was looking through it and I couldn't figure out how to do it.
Well,
and then I was thinking that we didn't have to do that. I thought we had to find the...
Yeah. No, you didn't. You should know how to do that because the pH is equal to the negative log of the hydrogen ion concentration. Mm-hmm. So that means that the hydrogen ion concentration is equal to 10 to the minus pH. So if you wrote on an exam 10 to the minus 3.2, you would have the answer. You're not going to calculate it, but you have to write down what you would do.
That makes a whole lot more sense.
Right?
I was looking at the book and I couldn't figure out how they were doing it.
Right. So in other words, you need to know how to write that down.
Okay.
Right. Okay.
Cool.
Makes
Thank
sense?
you. Yeah.
Clear as mud? Hi, what's up?
Are you posting a study guide or something like that online?
I haven't. I might be able to do that. I can't promise anything, but you think that would help?
Yeah, just because it's like a lot of information we don't really know what to pay attention.
Well, what I tell students too is look at the highlights because they're sort of daily study guides are what they are.
Yeah.
All right. I'll look and see if I can put some together, but I think for the most part, the highlights are really that already.
Okay.
So that's why I say focus on the study guides. Okay.
Or on the highlights.
Whatever we call them. You can see it's the end of the day for me too. So yeah.
Okay.
Okay.
And then just let us know about when the review session will be.
I will do that.
Okay.
Sure. Okay.
Thank you.
Take care, guys. Yep.